for organizing this very nice conference and nice meeting location. And my compliments to every one of you that you're still here. It will be soon that you're released to the dinner break. And before that, I would like to present, or Ilona and I, we would like together present 4 projects or 3 projects that we finished actually this year on GRIN-related disorders, and one that will keep us busy in the time to come, that is presented by Ilona. But I try to wrap up the first 3 projects quickly. The first one is on memantine. I believe everyone, or most of you, will know that memantine has been the first precision treatment in GRIN-related disorders. It has been published in 2014 by the group from Atlanta, from Emory University, showing that there was a significant treatment response in an individual with a GRIN2A gain-of-function variant. And ever since that, there were numerous individuals that had been treated with memantine. Several have been published. Some of them have benefited as well. Some others have not responded at all, and yet others have worsened under memantine treatment. So in the end, the picture was quite blurry, and we didn't know, is memantine a good compound or is it not? So what we did was we collected all individuals that we could get. There were 14 published individuals in the literature, and we also collected 21 novel cases from the registry, so 35 altogether. And first thing, we found that actually only 21 out of these 35 individuals had pathogenic gain-of-function variants. There were others that had not gain-of-function variants. There were even some individuals that had not even pathogenic variants, so that was actually not considered a GRIN-related disorder. And given this, you will not be surprised that, of course, some individuals will not respond, or maybe even worsen. So what we found was that, can we see? No, we can't. We do it that way, that if we only take the 21 individuals that had pathogenic gain-of-function variants in any of the GRIN genes, then more than half of these individuals improved with regard to behavior. Some of them had no changes, and a minority of them we had no information. But there was no one that worsened. Same thing for development. A larger fraction improved, almost half of the patients had no changes, and a third of the patients we didn't have any information on developmental outcome measures. And seizure frequency, you can see here that a quarter of the patients had a decreased seizure frequency, and also here actually quite impressive decreases. It was a bit similar to the responses that we have seen in the rajaprodil trial, I believe. Several individuals had no response. Actually, 3 individuals also worsened, and some others we had no information. So altogether, we actually can say if memantine is applied in GRIN-related disorders with pathogenic gain-of-function variants, then it really is a helpful drug. A surprising or a nice aspect is that there were actually not really any relevant side effects. And also, in those individuals that had null variants or loss-of-function variants, the application of memantine did not lead to a worsening of the situation. So it was tolerated well, even though we would think we should worsen from a pathologic or pathomechanistic perspective. But that's not the case, and that might be also relevant and interesting for the rajaprodil trial, where rajaprodil is going to be tested in loss-of-function cases as well. And lastly, and we also found that the distance to the channel pore is relevant for the treatment response. The further away, the better the outcome of the patient. The second study that we finished, so the last one is just being in revision in epilepsia, so we hope that it will be published by the end of this year. The next one is on a topic of GRIN2A and tumors. Maybe some of you know that there was an old previous publication from 2011 where exome sequencing identifies GRIN2A as frequently mutated in melanoma. 2010 was the first time that GRIN2A actually had been published at all in neurodevelopmental disorders. So this was a very early study. Exome sequencing was not long around by then. And I was always puzzled by this publication, and I thought, does it have anything to do with tumors? We had no evidence in our registry that individuals with GRIN2A actually have a predisposition to tumors or to cancer. So I always had doubts on this. So what we did now was we actually just did some statistical evaluations, and we checked the GNOMAT database, which is a large population database with millions, hundreds, thousands of healthy individuals. We also checked the COSMIC database, that is a database containing all sorts of genetic variation that ever had been found in any tumors and cancers. And we also checked ClinVar, which is a database collecting all variants that had been found in individuals with whatever disease, for example, also GRIN-related disorders. And what we found was that the distribution of the variants, depending on the cohort, was different. So there was, let's look at the middle one, there was a peak, a clustering of missense variants in individuals from ClinVar, which is not surprising because pathogenic variants that are also reported in ClinVar will mostly affect the functional domains that are located in the central part of the gene. Pathogenic variants in the C-terminal part or in the amino-terminal part are rare or almost absent, but vice versa. You can see here a slight increase of variants in the healthy populations in the amino-terminal part and in the C-terminal part as well. So there's clustering of these variants, a bit mirroring. And you can see that the variants found in the COSMIC database are actually evenly distributed along the gene. And if we look at the null variants, we can see pathogenic null variants are only been detected in the first part of the gene. In the last part of the gene, they are mostly found in GNOMAT because they do not matter too much. They will not cause a disease if a null variant is located in the distal part of the gene. And here also again, actually the variants in COSMIC have an even distribution along the gene. There's no pattern, and if there's no pattern, then it's actually a random distribution, and there's nothing that actually supports the theory that GRIN2A variants have anything to do with tumor or cancer. So that's actually good news, and still in our registry we don't have any evidence for cancer that is that would be prevalent in GRIN-related disorders. So we actually can say this publication is a bit outdated and nothing to worry about. And the last one was actually spoiled by Nicolas Beckjo half an hour ago. No, that's completely fine. That's a very novel situation. So it was published on Tuesday. I've never thought that anyone would have read it by Saturday and even presented it on a meeting, but I'm really impressed. Anyway, to make it short, we were actually prompted by this publication also from 2011, so also quite old, where it was suggested that GRIN variants, particular variants in GRIN2A, but also one or two variants in 2B, were associated with autism spectrum disorders and schizophrenia. But going back to this publication, actually the GRIN2A variants reported there are not pathogenic. But ever since that, there were other studies, however, linking GRIN2A and schizophrenia or mental disorders or psychiatric disorders. So what we did was going to the registry and and checking whether or not any individual with GRIN2A had actually a psychiatric disorder. And we found what you've already had seen in Nicola's talk, that particularly individuals with GRIN2A null variants have mental disorders. Those with missense variants, they usually do not have mental disorders. And if you look at the disorders individually, then you can see, okay, anxiety disorders, if you read this graph, then it means by the age of around 15 years, you will have a 20% likelihood, so 1 out of 5 of the child of having been diagnosed with an anxiety disorder. Almost as much for mood disorders, a bit less, but still relevantly high for psychotic disorders, but only for those individuals with GRIN2A null variants. And and we just published that. And that's the 3 studies we finished, and now it's Ilona's turn to present you the study we are currently working on, and I hand over to you.
Thank you. Yeah. And thank you very much again for inviting us and for changing the program last minute for us. Finally, due to the patent issue, I'm allowed to talk about our approach, the strategy, and our results of a single cell overexpressing model with ASOs. And I'm not the only one. We are an international team. Johannes, you've seen before, and then Steve Tranelis, you may all know, and Matthias Wagner, another geneticist from Munich, and we are working together. We are an inventor team. So it's a team effort. Before I start explaining our strategy, I like to point out 3 conditions that made our approach happen in the first place. So the portal and the registry, as well as the awesome cooperation and collaboration with you, with the parents and with the families, with the clinicians, allows us access to a large number of individuals for the potential trial participation. And second, it's the right time for ASOs. When we look at FDA approvals for ASOs, they are getting more and more, and also for neurodevelopmental disorders, for example, Dravet syndrome, we've heard about it before, or Angelman syndrome. So what are ASOs exactly, and how does they work? I like to explain you shortly. So ASOs are short oligonucleotides that can bind to the RNA in a target-specific manner and ultimately modify protein expression. So in the end, ASOs are not gene therapies because they do not alter the DNA, they target the RNA, and therefore they are a completely different type of mechanism. Third, GRIN1 is a perfect candidate for ASO because GRIN1 haploinsufficiency is not pathogenic. We know that missense variants in GRIN1 can lead to a gain or a loss of function of the NMDA receptor function. And regarding the genetic variant itself, we can call it gain or loss of function or dominant negative. This is kind of a genetic view. You may like more the word loss of function, but on a genetic perspective, it's a dominant negative variant, leading to a loss of receptor function. But truncating stop variants or frameshift variants are very frequently found in the general population, and therefore having only half of the receptor is not disease-causing. That's why we think GRIN1 is a perfect candidate for ASO, or to start with ASO in GRIN-related disorders. Yeah, therefore we sat together and developed 2 strategies for GRIN-related disorders. The first strategy targets all gain-of-function variants and aims to reduce the total expression of NMDA receptor protein by using gapmer ASOs, or we call it knockdown ASO, that binds to the most favorable and accessible regions within the GRIN1 coding sequence. Non-selective here means that the ASO binds somewhere in the gene and not to the specific mutation or a SNP. And this strategy would turn a hyperactivated cellular state into a more physiological neuronal activity. And again, GRIN1 haploinsufficiency is not pathogenic, so therefore this strategy may work for gain-of-function variants. The second strategy targets all heterozygous missense variants, so both the gain-of-function and the dominant negative variants. When the respective individual is heterozygous for a specific SNP. And a SNP is a single nucleotide polymorphism, and they differ between individuals, but they are very common, or at least in 1% of the population. And yeah, so this is an overview where we are now. We designed the ASO, we screened some ASOs, we did in vitro efficacy tests, and we submitted a patent application. So just to make sure we are in a very early stage of a preclinical stage. And in the next slides, I'd like to show you what we did in the in vitro efficacy test. So we injected the ASO and GRIN1 RNA into a frog oocyte. We incubated the oocyte, and then we compared wild type and the oocytes that had the ASO as well. We did a 2 electrovoltage clamp recording and measured the current size. And what you can see here is a picture I recorded, and you see that there's a current size up to 1,270. And here in this left figure, you see the current size. So the current size in purple, you see a current size from the wild type you've seen in the previous slide. This is the wild type current size, and the reduction of the current size means the ASO is working. So we designed 5 non-selective ASO, the strategy 1 I explained, and they differ in size and target region. And we tested them in the frog oocyte and measured the current size compared to the wild type receptor. And the non-selective ASO differ in effectiveness. So you see E1, E7, E8, they are all ASOs, and they differ in effectiveness. E8 and E13 are most effective, and especially E13 is the one that works best. Then we tested the E8 and the E13 in different concentrations to see if it's concentration dependent, and we saw that it is dose dependent, and E13 reduces the current size more efficiently than E8. We just compared these 2 ASOs. And you've seen this slide before. We also tested the second strategy, and where the ASO binds directly to the SNP, or to the wild type SNP. So you have 2 allele, one with a SNP, one with a wild type, and we designed 2 ASO, one that binds to the SNP and one to the wild type SNP. So what you can see in the left is that the SNP ASO also worked very well and also concentration dependent. And here in higher concentration, we observed a lower current size. And on the right, we wanted to see if the SNP ASO also binds to the wild type SNP, what shouldn't happen, but we know from other ASO studies that sometimes one nucleotide change is difficult to target. So we found out that only in very high concentrations the ASO also binds to the wild type SNP and reduces the current size significantly, so therefore we call the ASO allele selective and not allele specific. And we talked to other ASO guys and showed them our results, and they said this is what they also observed. So this is yeah, they said they are very good results. Sometimes also in a 0.1 concentration, they have up to 50% reduction. So yeah, they encourage us. It's very good results. But that's it. So that's all I can show you. That's all we have, and we are working very hard on the next steps. But what we discussed yesterday, the hurdles are high, so we need time, man and women power, and of course money. So I hope I can present you more next year. But yeah, we are working on it.